Well, I'm David Menton, and I'm here today to share a favorite topic of mine, and that's the development of a baby in the womb. Only we're only going to talk about the first week. So many things happen in the first week that are marvelous. We could just stay there. I will kind of go to the end when the baby's delivered. But let's dig in. I call this Fearfully and Wonderfully Made. I stole the title. I stole the title from Scripture. And I guess it's okay to steal from the Bible as long as you're going to share with people. Well, here's an age-old question for you. I bet you've all thought of it at least a few times. When does life begin? It's actually not a very precise question because life, the uterus itself is alive. The nail bed for our fingernails is alive. Our hair follicles are living. I guess what we're really asking is, when does a human life, an entire human life, begin? But I think even there, God created humans on the sixth day of creation. And it's been a continuum ever since. People begat people. If this was ever stopped, you'd never get it started again. So we could say, in a sense, that human life began on the sixth day of creation. But then what about each individual life? To me, the best question is not so much when does life begin in the womb, or even when does human life begin in the womb, but also to ask the question, when does a person begin? What's a person? Somebody recognized by God as a person, a unique person, different from everyone else. And that is something that is pretty amazing. Let's look into that. Jeremiah chapter 1, verse 5. Boy, this is a verse to keep you up at night. Before, God says to Jeremiah, "Before I formed you in the womb, I knew you. Before you were born, I sanctified you. I ordained you a prophet to the nations." Well, think of that. Before the worlds were formed, the Lord knew Jeremiah. He knew us. So our awareness to Him was way back before any of us can even begin to understand. We can understand when the baby first appears in the womb, and that's what we're going to focus on here. Psalm 139 tells us a little bit more about the development of the baby in the womb. It has some interesting names for the womb. There we read, "For you," that is God, "formed my inward parts. You covered me." Another word for that is knitted me, like knitting. "You covered me in my mother's womb. I will praise you, for I am," here it is, "fearfully and wonderfully made." That's where I got the title. And then this last phrase, "Marvelous are your works, and my soul knows very well." You know what that's really saying? God's works are so marvelous that it's just plain obvious. I'm amused when I get on Facebook or the internet and I see atheists arguing, insisting that there's not even the slightest trace of intelligent design in the human eye. No, the Lord says it's obvious. We know it very well. We know it in our soul that there's a creator. Psalm 139 goes on to give us more specifics. It says, "My frame," that means not just our skeleton, but really all the parts of our body. "My frame was not hidden from you," was not hidden from God, "when I was made in the secret." King James has secret place. This is New King James. "And I was skillfully wrought in the lowest parts of the earth." That's kind of a euphemism for the uterus, sort of like putting seed in a soil. Now, verse 16, to me, is the most powerful pro-life argument that anyone could ever have. Think of it. It says, "Your eyes," that's God's eyes, "your eyes saw my substance, being yet unformed, and in your book they all were written, the days fashioned for me, when as yet there was none of them." Do you get the point here? God watches the developing child. He watches as it develops in the womb. So let's look at that marvelous organ of the body, the womb that the lady so proudly bear. Here it is. It sits low in the pelvis. It's held in place by a broad ligament on each side, here and here. And that broad ligament has a kind of a clever name. It's called the broad ligament, as simple as that. The uterus is a smooth muscle. It's the kind of muscle over which we normally don't have control. And coming out of the top of the uterus are two ducts or tubes called the oviducts. You see them up here. Another name for them are the fallopian tubes, or the either name will suffice. Oviduct simply means an egg duct. And then on either side of the uterus, there's a little structure here and over here. And they are the ovaries, where the eggs reside. And these ovaries are about the size of an almond nut, to give you an idea of size. So you'd have no trouble seeing them without a microscope. And the oviduct comes down the end of it, very close to the ovaries, you'll notice there. And on the end of the oviduct is a funnel. Do I have to tell you? It's called the funnel, only in Latin. Infundibulum, it means a funnel. And you know what funnels do. Funnels catch things we might otherwise miss. And that's exactly what the oviduct does. It actually comes down in parks, sits right on the ovary, and moves around until it finds the spot where the egg is going to be released. So it doesn't take any chances on catching that egg. It moves right up. It has like little fingers in the end called fimbria that help in this. In any event, the egg will be released from the ovary, will go into the oviduct, and this will happen if a lady's regular every 28 days. The ovaries are courteous. They take turns. 1 month is one, and then the other, back and forth. It's interesting, if one of the ovaries is damaged so that it no longer functions or it's lost altogether, the remaining ovary has a considerable good sense to go ahead and ovulate every month. We call this compensation. The body's just full of things that compensate like that when necessary. Let's move on here. Let's take a close-up look at the ovaries. They say it's about the size of an almond nut. If we made a section through it, magnified it, it would look sort of like that. When a baby girl's born, when she first comes into this world, she has all the eggs she's ever going to have in her whole life. That's about 1 million. Apparently the Lord thought 1 million kids would be enough for anyone, huh? Obviously not all those eggs are going to be used for fertilization, but think of it, all the eggs are there at birth of a little girl. And when she reaches sexual maturity, they begin to mature. The eggs are inside of a structure called a follicle. So the egg is invested by cells, a follicular structure. And the follicle begins to grow in size, not the egg, but the cells around the follicle. And this follicle eventually builds up fluid in it. We sometimes call that an antral follicle. And the fluid increases and increases until the follicle just literally bulges out at the surface of the ovary. At the appropriate time, a little hole will develop on the end here, and the egg, which is back here, will be ejected out through that hole into the waiting funnel of the oviduct. This picture, or part of the picture down here, shows the egg being ejected, and every 28 days, typically about 30 to 40 of these eggs start to mature. And this cohort of 30 or 40 breaks down progressively until you end up with only one egg. Sometimes you have more than one, then you can have multiple births, twins. But typically just one egg. The other 30 or 40, whatever number there was, go into what we call atresia. They break down. This is not abortion. This is way before fertilization. This egg is released into the oviduct. We'll follow it later. Right now, let's look at what's left of these follicle cells that were around the egg. You see you have a rim here. They form an endocrine gland that's yellow in color, so it's called the corpus luteum, which means the yellow body. And this endocrine gland produces a hormone, actually a couple, but an important one is progesterone. This hormone will cause the uterine wall to develop glands that will provide nourishment for the conceptus when it first arrives at the uterus. If fertilization, or if pregnancy doesn't occur, fertilization fails, then a signal goes out to this gland to break down. It's not needed. We don't need to prepare the uterine wall to receive the baby. So When that signal is given, it breaks down and forms connective tissue. Basically forms a scar. And if you have any scars, you know they're usually poorly vascularized. I have a scar right here where I walked through a glass door many, many years ago. It's still there. It hasn't gone anywhere in about 50 years, 60 years. So, yeah, scars are pesky things. They just don't go away. And think of it. Every 28 days, every time a corpus luteum breaks down, a scar is formed. This scar is called the corpus albicans, which means the white body. And these scars are big enough to see with the eye. You do not need a microscope. You could take a microscope slide, hold it at arm's length, looking at the ovary the size of an almond nut, and you could see these scars. Every 28 days, you get another one. Anyone see a problem coming up here? How many months would it take before the ovary was a solid mass of scar tissue? Oh boy, that would spoil everything, wouldn't it? Well, what happens is just amazing. It's the only place in the body I know of where scars are completely dissolved. Boy, the plastic surgeons would like to pick up on that trick, I'll bet. Scars are dissolved away, and if this didn't happen, the ovary would become a mass of scar tissue eventually. Well, let's follow the egg in its path within the oviduct and uterus. Here we have a cutaway showing the oviduct coming out of the uterus, and the funnel applied to the surface of the ovary. And in this area right here, the egg will be released, and the egg will be propelled down the oviduct with its cells that are around it, its follicular cells. And it'll go all the way down here and implant in the dorsal wall, typically, of the uterus. And that takes about a week for that whole action from the time it is fertilized and it gets down there. What causes the egg to move down the oviduct? Well, that's an interesting thing. If you look where that little green box is, we're going to magnify that area, and it won't just be a picture, it'll be a real oviduct, and it'll show the lining cells of the oviduct. Here it is. These are called ciliated cells. They have little hairs. You can see the hairs at the top. Now, when I call them hairs, don't confuse them with human hair. These are just vastly smaller than a human hair. In fact, these hairs are so tiny you can't see them in a light microscope as an individual. You can see them collectively, the way you can see hair in somebody's head maybe a block away. But if they held up one hair, you couldn't see it. So the hairs are actually thinner than they even look here. And these little hairs beat, and the beating of the hairs, the movement, propels the egg down the oviduct, or it could be a fertilized egg. So how are they going to beat? Be an engineer for a bit now. How will you have these little hairs beat? They don't beat like an oar. They don't go like this. They're like a whip. They go like this. But what kind of a pattern? Are they all just going to beat to their own little drummer? All these little hairs going like this, millions and millions of them? No, that wouldn't move the egg or conceptus down the oviduct. If they all move like window wipers in a car, all together, that wouldn't work either. There's only one movement we know of that can successfully move this fertilized egg or conceptus down the oviduct. And it's a movement most of you are familiar with. In fact, you've tried to do it with your own arms. That movement is called the wave. You ever do the wave? You get a whole baseball or basketball stadium full of fans all trying to do the wave. It takes a lot of coordination. You can imagine millions and millions and millions of these little hairs lining the oviduct, all beating in this particular rhythm. The technical name for this is it's a metachronal rhythm, but it's basically the wave. And that's what will propel the egg down the oviduct. Well, we're going to look at about 9 stages here. Here is the egg when it first arrives in the oviduct. And we're going to make a cross-section across the oviduct, sort of like this. And we have a very lucky cross-section here. We actually hit the egg in the oviduct. This is a section that I saw when I was teaching in a medical school. One of my students had this slide. It's a very lucky slide. So he didn't have it very long. Once I saw it, I gave him another slide, and I took that one. It's good to be the king. So anyway, we caught the egg right in the oviduct. This is not a human. It's I believe, a rat. But it would look very similar. to the situation in the human. The cilia are all around the edge here. That's where they bead. And then here's the egg. This is one cell. This is a huge cell. A typical cell is more like these little ones that are around the outside here. They're called the cumulus oophorus. You see the Latin name there? You know what that means? It means the cells that accumulate around the egg. You probably think, boy, it's a pity to have the egg so messy with all these sticky cells still clinging to the egg. You should have cleaned it up. Well, if you cleaned it up and took those sticky cells off, the egg would not go down the oviduct. It's been shown experimentally in rabbits that if you clean off the egg just spins in the oviduct. It doesn't move down. Or if you make a glass bead the size of an egg, it won't go down the oviduct. By the way, in case you're wondering just how big is a human egg like that, I mean, could you see it with your eye? Yeah, with good vision, you would see it with your eye. It's about the size of a grain of salt. So big for a single cell. Let's crank up the power a little bit. We find out that this section was even more fortuitous in catching the egg in the oviduct. It also caught the chromosomes. Look how tiny the chromosomes are. They're being pointed to there and labeled chromosomes. Look how tiny they are compared to the size of the egg. Why is the egg so big? The egg is big because it's full of yolk. Yeah, a lot of people don't even realize where you come from eggs. They're not eggs that are laid outside the body. They stay inside the mother's body. And we do use that yolk until other forms of nutrition become available, like the wall of the uterus and finally the placenta. Cells have to divide twice to make a germ cell. You see, we have 46 chromosomes, and if the sperm cell has 46 and if the egg had 46, the two would get together, you'd have 92 chromosomes. And in the next generation, you double again. So the germ cells have to have their number of chromosomes before they fuse together. And this egg cell has gone through one division and parked at metaphase of the second division, and this is a special division called meiosis. Basically, it duplicates the DNA once, but divides twice. So we get down to the 23 chromosomes instead of 46. So when you get 23 and 23 come together, you're back up to the full complement of chromosomes again. Notice there's a clear zone around the egg. Do you see it out here? It's labeled zona pellucida. You know what zona pellucida means in Latin? It means a clear zone. And that's our eggshell. I'll bet you didn't know we had an eggshell. Admit it, you didn't know that. Yeah, we had an eggshell. It's not a mineral shell like you see in a chicken. It's more like a gel, like tough jello. And that shell turns out to be important, as you'll see in a bit. Well, let's move on here. Here we are 12 to 24 hours after ovulation. This is the period of time when fertilization typically will occur. The sperm have to be in the oviduct for about 12 hours to be capacitated. Oh yeah, you have a step there where certain chemicals in the female reproductive system modify the surface membrane of the egg. And if you don't have this capacitation process, sperm are incapable of fertilizing an egg. So all kinds of detailed, complex things going on here. Typically, we're talking about 150 million sperm trying to find their way up the uterus and into the oviduct to get to one female germ cell, the egg. Think of it, 150 million male germ cells to find one female germ cell. I'm guessing the reason for that number is that males just don't ask directions. That's the problem. Typically about a dozen sperm cells will find their way to the egg, and only one can be allowed to fertilize the egg. Here we are at the egg. We see 3 or 4 sperm cells there in the periphery. And right away, the male germ cell has a challenge to get through the cumulus. That sticky stuff that is produced by the cumulus is impenetrable, except a special enzyme is produced that digests through it. Then you get to the zona pellucida, and it's not going to get through the zona pellucida either without another enzyme being produced, which digests its way through the zona pellucida, or the eggshell. And finally, the sperm cell gets down and touches the egg cell membrane. And when it touches the egg cell membrane, there's a quick biochemical inventory that takes place. Basically, it goes something like this. Are you really a male germ cell, a sperm cell, because if you're not, they will not gain entrance. Are you the same species? Well, this is the end of evolution. If it's not the same species or the same kind, that germ cell is not getting in. And then, have you been capacitated? Did you get all the chemical modifications to your membrane? And if the answer to all of these, the biochemical answer, is affirmative, or yes, then it's basically come on in, and the sperm enters the egg. Now, when the first sperm touches the egg membrane, there's a special reaction called the cortical reaction. Cortical granules kind of explode and produce an enzyme, ovoperoxidase, that hardens the shell of the zona pellucida, and that keeps all other sperm from getting in. If more than one sperm manages to get in, it's called polyspermy, and when that happens, everything breaks down immediately. This does not go through and end up as fertilization. This breaks down before fertilization is complete. Well, let's take a look at what the egg cell looks like when the sperm has entered into the egg. And here is a picture showing a human egg cell. And notice there's two little balls inside. Those are called pronuclei. One of them has the chromosomes or the chromatin material from the mother, and one has the chromosome material from the father, each basically 23 chromosomes. Those two pronuclei are going to fuse and form one nucleus, one bigger nucleus. And the chromosomes get together, and that is the moment of fertilization or conception, where we have a new child. This results in a genetically distinct human being that's unlike every other human being that's ever existed on Earth. They're not identical, they're a little different. We call that fertilization then. Another name for it is conception. But definitions are getting played with, let's say, by the scientific and medical community. I was a consultant for several editions of Stedman's Medical Dictionary. I did all of the terms relating, or was in charge of the terms relating to microscopic structures, histology. I was working for the dictionary in the 26th edition in 1995, and conception was defined this way: the act of conceiving or becoming pregnant, fertilization of the oocyte or ovum by a spermatozoon to form a viable zygote. That's the name of the fertilized egg properly, a zygote. In 2000, that was the year I retired from the medical school. I also retired from participating in Stedman's dictionary. In 2000, the 27th edition came out, and the definition of conception was completely changed. This is the old one here. They took out that part and they put in this. Now conception, which is supposed to be a synonym for fertilization, is defined as the implantation of the blastocyte into the endometrium. This is a process which begins about a week after fertilization and is completed about 9 or 10 days after fertilization. They're buying all that time and saying that basically conception hasn't occurred yet. And why did they change the definition like that? I'm going to take a guess on this one. I think it's so they could be arguing when Plan B came out, a form of birth control, that it doesn't interfere with conception. Definitely can interfere with fertilization, but they say it doesn't interfere with conception. My understanding is the arguments have gone on and the definition has been changed back and forth a few times since I retired. Well, let's move on to 30 hours after fertilization. This is when a baby becomes a two-cell human being. Think of it, 2 cells. It still has a shell around it. It's shown in red there. Have you ever seen a 2-celled human being? I have a picture of one right here. There it is. Each has a nucleus. Each has cytoplasm. And notice the eggshell, the zona pellucida, is still around them. Now we're going to find out why this zona pellucida is important. You see these early cleavages when the cell is dividing 2, 4, 8, 16, etc. These cells don't stick together very well. The sorts of things that hold the cells of our body together, like desmosomes, tight junctions, gap junctions, what have you, these haven't really developed yet. intercellular cements that hold cells together haven't really fully developed. And so if you don't have a shell holding the cells together, they can separate. And if they separate, what happens? Oh my goodness, you could lose some sleep over this one. Those two cells you're looking at right there, if they separated, could end up producing this right here. Identical twins. That's how we get identical twins. Think of it, it's a clone. It's one egg, one sperm, got together, formed the zygote or conceptus, and now it's starting to divide 2, 4, 8. But if the cells separate, each of them can form a baby. I don't know what the upper limit is. I've seen identical triplets. And by the way, identical twins really aren't identical. They have different fingerprints. Think of it, the same genes, but different fingerprints. So there's a lot of things going on besides just genes in the body. But think of two such lovely children coming from two cells. Well, let's move on from 30 hours to 3 days after fertilization. I want you to notice something kind of interesting here. There's several cells. As I say, it goes 2, 4, 8, 16, 32 as it divides. Notice, even though the cells are dividing, this structure is not getting any bigger. We call it a morula because that's the scientific name for a raspberry, morula. And these structures look lumpy, sort of like a raspberry, so that's their scientific name. It's a baby, but we give it the scientific name morula. But it doesn't get any bigger. And if you think about it, it can't, because it's still inside this shell. And if it got bigger, it might clog the oviduct as well. So these early divisions are like cutting up a pie. You can cut it into 2 sections, 4 down the line. Pretty much the same size pie, isn't it? Well, here we are at 4 days, and we're just entering the oviduct. Let's take a look at the morula here. This is one sitting on the point of a pin. That's how big all of you were at one time. Sitting on the point of a pin. We hear people say, are you going to be concerned about that little tiny mass of cells, that's an abortion if we destroy that? Is God concerned about us that are small, that are tiny? Boy, here's a beautiful verse from Job. Job asked God, what is man that you should exalt him, that you should set your heart on him, that you should visit him every morning and test him every moment? Who are we that God should send his son to take the hit we should have taken, the punishment for our sins, for the sins of humankind, to take it upon his own son? What kind of love is that? We can't even begin to fathom it. But that's the love that is God. So yeah, God even loves that little morula. It's a tiny baby. Well, here we are at about 4.5 to 5 days. The structure now is no longer called a morula, it's called a blastocyst. What's the difference? A morula was a solid ball of cells forming the baby. The blastocyst has some fluid in it. It's a hollow ball. Well, let's take a look at a real human being in the blastocyst stage right here. There's 107 cells in this. Think 3D. This is a ball. The area in the middle, there's a liquid in here, or fluid. Here's a layer of cells out here, and then it gets thicker right here. You're probably wondering, where is the baby here? I'll show you where the baby is. The baby's right there. That part is called the inner cell mass. That is the developing baby. You might wonder, well, what are all these other cells doing? What do they do for a living out here? Oh, you've forgotten about something. And we better start thinking about it, or we're never going to get this baby born. They're called trophoblast cells. When they're separate like this, we call them cytotrophoblast cells. And they have the ability to fuse together. So 2 cytotrophoblast cells touch one another, and their membranes can fuse, and now we have a bigger cell with 2 nuclei. Another cell fuses, we have a bigger cell with 3 nuclei. This goes on and on and on, until we form a cell so big that if you could flatten it out, it'd be the size of a living room area rug, 8 by 11 feet, called a syncytial trophoblast. That cell, singular as far as we know, covers the whole surface of the placenta. And the syncytial trophoblast has a very interesting property. They're highly invasive. It allows them to invade into the uterus of the mother. something that would normally never be permitted. We don't allow foreign tissue to enter our body fights it. And it would fight this blastocyst if the immune system wasn't adjusted in a way that allows it to enter. So trophoblast, that's the way we spell it, cytotrophoblast or syncytia. We'll learn more about that as we go along. At 5.5 to 6 days, we begin to implant. So the new definition of conception would say that now we're getting involved with conception as we implant. But fertilization occurred way back here. But the defined conception is something that's happening way over here, 5 days later, and this isn't complete until about 9 or 10 days. So that allows them to say that this pill or that pill doesn't interfere with conception when it does interfere with the development of the baby. Well, let's take a look at this at a little higher power. There's this blastocyst. It has a liquid-filled center. You can see the blue there is where the baby is developing, the inner cell mass. These trophoblast cells fuse together and form like little tongues, and these penetrate right through the endometrium of the mother, through the surface layer into the connective tissue underneath. Here the process has gone a little further. We're showing one of the uterine glands. This produces a kind of a sugar-rich material that the baby, the developing baby, will use for nutrition until it can develop its placenta. There are also a lot of blood vessels here in the uterus that will provide nutrition through the placenta when it develops. But the placenta is just developing here. Notice placenta is developing even as baby is developing over here. Here we're at 14 days. A lot of cells have fused together to form what we show as a pink blob here of material. Blood flows in spaces in here called lacuna. This is an amazing blood supply. The syncytial trophoblast erodes the blood vessels, just like cutting the end of a garden hose off. That blood would be everywhere inside the uterus were it not for the fact that the placenta is sealed all around the edge here, and the blood stays in this area. So baby's blood and mother's blood will come into close association without ever mixing. Well, this is what the placenta looks like from the baby's side. The uterus should be on the far side. We see the umbilical cord coming out of there. When I look at the umbilical cord, I know there's a crater. There's a tissue in there that doesn't occur anywhere else. It's called the Wharton's jelly. And it's jelly, all right. It's just springy. And why is this so? Well, you have 3 blood vessels in there supplying blood for the baby, going to the mother to pick up nutrition and oxygen, coming back to the baby. And these vessels don't dare kink. The baby is kicking around at a certain point, and this Wharton's jelly is so springy it keeps the vessels from kinking under all but extreme conditions. What does the placenta do for a living? It's easier to tell you what it doesn't do. It's not a brain, it's not a central nervous system, so it doesn't serve any of those functions. And it's not a heart. The baby has to pump its own blood. And it doesn't make blood, the baby has to make its own blood. This is why when you look at an embryo, you see this big bulge coming out of the front. It's called the cardiac prominence. The heart has to develop early. All the other organs of the body, the placenta, with the help of the mother, takes over those functions until the organ in the baby is developed to the point where it's functional. So what are all these functions the placenta is doing? Here's a short list. The placenta is essentially the baby's lung. The oxygen is coming from the mother's lung, but through the placenta. And this is the baby's source of air. And because the baby itself is under liquid, right? So it can't breathe on its own when it's under a liquid. Actually, there's a dreadful disorder where the lung fails to develop in rare cases. And the placenta is so efficient that a baby can go all the way to term and be delivered with no lungs. But as soon as it's separated from the placenta, then of course it dies. The placenta is the liver. In fact, it's the whole digestive organ for the baby. It takes over all those functions. So it's the whole gastrointestinal tract. The placenta is really the whole urinary system. It's the baby's kidneys. The placenta is the whole endocrine system for the baby, provides all of the hormones and things that are necessary. And the placenta is a protector. What do we mean by that? It gets antibodies from the mother before the baby can make its own to ward off any kinds of problems that may occur. So quite an organ, huh? It serves for 9 months, and then when your baby's born, the placenta breaks down very quickly. It has almost no shelf life to it at all. Medical students, if they want to look at a placenta, have to pretty well see one from the delivery room. We don't even bother to try to fix them or preserve them, too fragile. Well, this is what the whole placenta looks like in a very diagrammatic way in cross-section. You see the vessel coming out of the top, that would be going to the baby's belly. And inside here are 3 blood vessels. There are 2 veins that we call the umbilical veins, and they're carrying oxygenated blood. Usually we think of veins as carrying unoxygenated blood, but we call a vessel a vein if blood is coming towards the heart. We call it an artery if it's going away. So this blood returning from the mother's placenta to the baby, which would be above this illustration here, this is oxygenated blood coming through these veins, and there's 2 of them because a baby has 2 legs. These vessels actually sprout off from arteries that are going down into the baby's leg, and the 2 converge on the umbilicus or belly button, as you might call it. And so there's 2 of these umbilical veins. And then there's just 1 placenta, so there's 1 umbilical artery, and the umbilical artery is right in there packaged with the umbilical veins. The surface down here is the uterus. All of this is the uterine wall. Remember, it's been eroded when the baby came in, or when it developed, it eroded this whole endometrial wall here. You can see blood vessels just ending freely, just like cutting off a garden hose. This blood then, under high pressure, comes in and fills this whole labyrinthine area here with mother's blood. Baby's blood is inside of little structures that look like a tree upside down. See the trunk here and the tree? Technical name for that is a cotyledon, but it really does look like a tree. It has a trunk with lots of branches out to little tiny twigs. The twigs are so fine they just look like fuzz. And baby's blood is sort of like the sap running in the tree inside of the cotyledon. Mother's blood is like the wind blowing through this little grove of trees. And all of the transition of gases and nutrients has to be across, I guess, what you might call the bark on the surface. And this surface is that cell I was telling you about earlier, the syncytial trophoblast. As far as we know, a single cell with millions and millions and millions of nuclei that covers this surface. So it's absolutely seamless barrier between mother's blood and baby's blood. As you approach term, all of the blood of the mother goes through that placenta every 10 minutes. That's a pretty fast blood flow. Now, here's the scary part. When the baby's born, it leaves the womb, and then the placenta comes as the afterbirth. That placenta unplugs from the uterine wall. And when it does that, there are dozens of arteries and veins truncated with blood flowing out of them at the rate of all of the mother's blood every 10 minutes. This means when the placenta unplugs, you might reasonably expect to get total insanguination of the mother in 10 minutes. This is, without a doubt, the worst wound any human being ever gets and lives to tell a story. What could be worse than having all the blood of your body drained in 10 minutes? Maybe if you didn't have a clotting mechanism, that would even be slightly worse, if anything could be worse. And in fact, the clotting mechanism is shut down in the placenta. So this is a serious wound. We've eliminated temporarily blood clotting, and we've pulled the placenta off, exposing truncated vessels. What kind of a plan does the Lord have to deal with this? Oh, this is so wonderful, I should charge you $5 a piece to tell you this. Every one of those vessels, every artery and vein, has a muscular sphincter, sort of like a purse string. And as soon as it's cut off, as the placenta breaks away, those purse strings, those muscular sphincters, close, just like a hemostat that a surgeon might use. And so instead of all of the blood of the mother being lost in 10 minutes, normal pregnancy, there might be a loss of about a cup of blood. How about that for a miracle? Got any evolutionists out there? How's dumb luck working for you on this one? How would you sneak up on this step by step? Well, this is sort of the whole flow pattern here. The blood is coming out almost like from a garden hose. The cotyledons look like you might be watering some little juniper tree or something. It sort of blasts right into them. And then the veins are sort of like drains in the floor that drain the blood out of the uterus again. So this is this whole blood flow mechanism. Well, here's the placenta at 40 days of development. You see the baby in the middle there. At this stage, this may surprise some of you, the placenta goes all the way around the baby. Think in 3D. It's a whole ball going around the baby, and the baby's inside. And look how fine the branches are. It looks like cotton candy. Where would a cotyledon, one of these trees I was talking, where would that be? It would be in a position sort of like there, showing in yellow. And there would be many of these. Typically in a placenta, as you approach term, there's about 20 cotyledons, more or less. If you magnify the very tip of one of those little branches from that cotyledon, looks like this. This is a picture I took, I think, 1 million years ago. In fact, I think it was one of the first scanning electron micrographs, showing this detail on the surface of the placental villus. The surface area is amplified with this little tiny microvilli that come off. So that's why it's estimated that the surface area of the placenta, which is, as far as we know, is a single cell, all fused from millions of separate cells, this surface area would be about the size of a living room area rug, 8 by 11 feet, if you could flatten it all out, rather than coating all these little trees. Then if we make a cross-section, say like that, across that tiny little microscopic villus, as they say, this is in a scanning electron microscope, so it's highly magnified. More than we could do with a light microscope. Well, we could do about that magnification. With a light microscope, we wouldn't see the detail on the surface. This is what the inside looks like in the microscope. And when we look inside the little villi, we can see that there are red blood cells in here, and there's some white blood cells, like this little cell here. They have a nucleus. So these are baby's red blood cells here and here. Here's one right here. Where's mom's red blood cell? Here's one out here. So baby's blood is always inside these little villi. Mom's blood is always like the wind going through these little trees outside. So this white blood cell belongs to mom, that red blood cell belongs to mom, and the two bloods are separated by this pink line. And notice this pink line is interrupted every once in a while with some blue structures. Those are the nuclei of the cells that have fused together to make this structure. They get pushed off into little clusters or piles to leave as much surface area available for the transmission of all the things that have to go across the placenta. So the baby's oxygen comes from there, it gets rid of its CO2, all of its nutrients are going to come from mother's blood. Across that barrier to baby's blood, there will be no mixing of the bloods, whatever. Baby has to make its own blood. And by the way, for the baby to make its own blood, it needs to make hemoglobin. To make hemoglobin, it needs iron. To make iron, it needs to get it from mother's blood, because that's its only source of iron. The problem is iron won't go across that barrier by itself. There's an enzyme, a protein that's 670 amino acids long, so it's a big one. It's a glycoprotein called transferrin. Transferrin picks up iron from mother's blood, gets it across the barrier, and releases it to baby's blood. If that was the only thing that was missing in your body, is that protein, transferrin, it wouldn't do you any good whatever else you had. You wouldn't live. Think of it, 100,000 proteins in our body, and this is one of them, transferrin. Well, here we are at 7 weeks of development. This is about the time when get to the end of the 7th week, we call it a fetus, going from the 7th week to the 8th week. And so we go from an embryo to a fetus. It's still a baby. It's been the same baby all along, just different names for different stages. And what's the difference between an embryo and a fetus? Well, basically, we call it a fetus when all of the basic organs are in place. They may not be entirely functional, but they're laid down. They need to mature a little more, but it's all present. So, for example, here at 7 weeks, this fetus has developing milk teeth. About 99% of its muscles have at least begun to develop and are in place. This little baby would have detectable brain activity, and when it reaches 8 weeks, we call that the fetal period. It remains called a fetus until it's born. You know, I want you to think about something. Jesus Christ did not come into this world as a 30-year-old itinerant preacher. Jesus Christ came into this world as a zygote, which is a fertilized egg. Jesus Christ was a morula when it looked like a raspberry. Jesus Christ went through the stage in Mary's womb where he was a blastocyst. Jesus Christ was an embryo. He was a fetus. He was a newborn. He died for all those kids too. He lived through all those stages. He experienced all that just as we did in our own development. They're all babies and he died for them all. Well, let's look at a 12-week baby. Here it is, 12-week fetus. What do we have in place at this time? This little child has fingerprints on its fingers. It has fingernails. It has facial expressions. And let me tell you something about facial expressions. It takes about 45 muscles to make all of our facial expressions. It's the only language that wasn't confounded at the Tower of Babel. You can go anywhere in the world. A smile is a smile, a frown is a frown. Happiness and sadness are identifiable. Think of it, facial expressions are already being shown by a baby, 12 weeks of development. This baby has fine hair on its face. It swallows amniotic fluid. It responds to touch. It grasps objects placed into its hand. You want real proof this is a baby? It sucks its thumb. How big is a 12-week fetus that has all of this capability? I have a model of a 12-week fetus right here. It's a little over 2 inches from crown to rump. And it's a human being, no question about it. Well, let's just jump over from 12 weeks. Let's jump to term, 9 months, full term. I have good news to report. The baby has done well. Everything is normal. It's in the right position, head down. We don't want a breech first delivery. The umbilical cord does not appear to have been kinked or branched. I mean, everything seems like it's great, except do I have bad news for you? Have you heard about the guy that built a boat in his basement and he didn't measure the size of the door? The baby has to get out through this opening in the pelvis. I have a couple of models of the human pelvis here. This one is a female pelvis, and this is called the pelvic inlet up here, and this is the pelvic outlet down at the bottom. Notice it's almost cylindrical. And even here, the baby's head would not be able to get through here, were it not for the fact that the joints between the sacrum, this part here of the pelvis, and the iliac blades out here, those two joints, the famous sacroiliac joints, they soften up. They don't dissolve altogether. They get so they wiggle. And then even where the pubic bones come together in front, this joint gets softened, and the whole pelvis opens to just allow the baby's head through. You have to turn it 90 degrees to fit. This is why males don't deliver the babies. Look at that pelvis. This is a male pelvis. Pelvic inlet doesn't look too bad, but the pelvic outlet, even by softening the joints, would never allow the passage of the baby through. Well, you weren't going to get through this lecture without seeing my grandson. He was born 2 months premature. And here he is in the neonatal intensive care unit. He lost some of his buddies and friends in the neonatal intensive care unit, but he made it. He's in college now, doing well. But it's a real battle to be born that young. Your lungs have a hard time getting adequate oxygen, and steps have to be taken to help that out. But every time I look at this picture, I think, now that's that's a baby at 7 months of development. It should be in the womb. He weighed 2 pounds, 4 ounces, by the way, in that picture. When I think of a 7-month-old fetus in the womb, I often think of John the Baptist. Remember when Mary and Elizabeth met? Elizabeth was pregnant with John the Baptist. Mary was pregnant with Jesus. And Elizabeth said to Mary, "For indeed, as soon as the voice of your greeting sounded in my ears, the babe leaped in my womb for joy." What was John the Baptist's joy? What is a 7-month developed baby in the womb waiting 2 months yet to be born? What does it know about joy? And what is its joy in? I see no other solution than the joy at hearing the sound of his Savior's mother's voice. What do babies know? Huh? What can they know? Well, it's not enough to be born once. You need to be born twice. In John chapter 3, verse 3 and 4, Jesus was talking to a man named Nicodemus, and Jesus said to him, "Most assuredly, I say to you, unless one is born again, he cannot see the kingdom of God." We've just talked about the biological birth, and it was pretty amazing, wasn't it? All the things that go on that have to be just right. Well, that first birth is nothing compared to the second birth. Because that second birth that Christ is describing is turning us around and sending us off in a whole new path, from dead in trespasses and sin to eternal life. Nicodemus said to him, "How can a man be born when he's old? Can he enter a second time into his mother's womb and be born?" That wouldn't even be birth if it were possible. You wouldn't call that birth if you were able to just crawl into the womb, crawl out again. But Nicodemus was confused. No, it's a thing that involves God's Word and the Holy Spirit, which in itself then changes the heart. And we're truly born again.